know somebody who has had a positive experience with cannabis sativa, not just CBD, just cannabis sativa. And I do this in a number of places when I speak. I speak on medicinal plants in various places in the world, and it's amazing how many hands go up. This is a little bit biased in here because you come from the industry, but people have this experience. And that experiential evidence is not founded and based on what we typically use as scientific method. Scientific method is based on mechanisms and hypotheses that get tested. But it is how we've experienced plant medicine for 5,000 years. So for over 20 years, actually it's going on 25 years, I've been out traveling the world, going to different remote communities and asking the people these questions about how do you heal? How do you use plants for nutrition, for a healthy lifestyle? What are those plants? What do they look like? How do you classify them? And we find out that people have been using plants for a long time for healing, and they know this knowledge of plants at a very detailed level. And that's pretty cool. It's pretty exciting because I've spent my life looking at plants, studying taxonomy. I'm a classic morphologist, classic botanist that crawls around in the woods, names new species of plants. Back in June, I was in the Amazon looking for a particular species in the Myristicaceae, which is the nutmeg family that's very bioactive. We had to go deep into Manaus, down a bunch of tributaries of the Amazon. This is the type of work that I do, crawling around looking at these plants. So when you meet people that have lived for generations and they understand plants at a very personal level, experiential level, it's quite profound for me. And I think we're coming to a time now, especially with social media, where this experiential evidence is coming back full circle. People now are sharing their experience with each other, which we do, and some of you put your hands up, you do that. It's like, oh, I gave this to my parents. I actually did that. My parents had sore joints. I grew up on a farm. They were very active all their lives. They moved off the farm, and they started putting on weight. They had sore knees and ankles that started to compound on themselves. And I thought, well, let's cook up some cannabis. So I took a mixture of low THC, around between 1% and 2%, and high CBD, it was between 12% and 14%. Cooked up the leaves in a crock pot with coconut oil, made a paste, and I got them to put it on their knees and their ankles. And they started, and I said, get out and walk. And they started walking. They went around the block, and I was there last Sunday. They're walking 4 or 5 kilometers a day. They lost 20, 30 pounds, and they're feeling great. And they kind of forgot what happened. But it's their experience, and they had this experience which is quite positive. Okay, break time. Okay, good. This is much better. So I teach first-year biology. I have 2,200 students, and it's a lot of fun. I get up there and share some of the experiences, and I get them to come out and work directly with plants. Here's how my storyline is going to go today, if you look up at the screen. For me, it all starts with a voucher. So I'm curator of an herbarium. Herbarium's a museum of plants. We press and mount plants. Those nutmegs that I collected from the jungles in Brazil, south of Manaus, will be pressed and mounted on samples that look like this. You can see there's a colorized code below it. That represents a DNA sequence for it. For that, it's called a DNA barcode, which is quite parochial. It's quite a small section of the genome. I'll talk about that a little bit later. So we collect a sample, we extract DNA, and we barcode it. So that's part of the story. There must be an archive sample that we go from, and that's a cannabis sativa sample. The second set is looking at chemistry. I look at a particular type of chemistry, metabolomics. And in metabolomics, I'm going to explain how it works in a nutshell and talk about why that is so important for the industry. Oh, lovely. Goodbye. And then lastly, I want to talk about supply chain management. I just came off a 2-week meeting tour throughout Europe. We started in Amsterdam, we moved to Geneva, we went into Germany, we went into London, and then over into France. And during that time, we were asking the same question about cannabis sativa. It's moving into edible markets, it's moving into food. And for the last 15 years, We've been working specifically in my research program to give tools to the industry for measuring GMP, quality assurance metrics for identity and purity, which is the law. You must do this. In the cannabis industry, you haven't had the background for chemistry and reference materials to be able to do these types of tests. They're often done in mass spec, HPLC. It doesn't exist right now. So in a number of these countries, we're talking about how do we actually do this? What's the limit of detection, limit of quantification? How do we actually measure CBD with precision and accuracy? In the States, I'm working with Focus Group. I'll talk more about it a little bit later. FDA, US Pharmacopoeia, National Institute of Standards. So I'm on their advisory boards to be able to figure out how can you make accurate tests in the industry so that we have a consistent product and we have a product that actually meets specifications and standards. This is new, fairly new for the industry. Here's my home university. Those aren't the university buildings. They're actually, those two buildings are my buildings. We've been very well funded, $150 million from the Canadian government to really do one thing, explore biodiversity and build libraries, genomic, metabolomic libraries. It's what we do. I'll talk about that in a few minutes. This is the True ID office. It's in the University of Guelph Research Park, and it's quite a busy place right now. So we take on cannabis. This is a new project that we just got funded. If you want to come up sometime and visit me, I have quite a large team. I have 80 scientists working for me. We're putting up a new building focused on cannabis. So for Canada, it'll be the Center for Cannabinoid Research. So I'm going to go back here. So here comes my first year biology lessons that I want to share with you. This is part of the education. So DNA is everywhere. Right now, in this room, DNA is floating off of your skin in something we call oligos. Short chains that are broken, that come off your skin, out of your cells, come out of your hair, and they're floating in the room, and they're settling on surfaces. And those forensic movies, when you see them going and they're swabbing up these little bits of oligos, and then you can amplify them and you can sequence them. And there's an algorithm. A certain amount of time that new master spends in this room, oligos are floating in the air. There's a certain concentration that are going to be found, and you can actually calculate how long I was in that room. And if there was a crime in here, if I walked off with this mic, which is like $1,500, I'd go like, yeah, new master's here. But nobody's going to do that. That is a concept that you need to think about always in GMP. Because these small bits of oligos are found everywhere. They come off the farm, they're in your manufacturing facilities, and when you start doing DNA testing, you need to be very cognizant and aware that this bits of DNA, small bits, are in all of your products. So if people start testing your products, and this is why I share it, and they go, you have contamination, be aware that that might be those small bits of DNA that are found from all over the place, because they float in the air. Even what you have for lunch is floating around in your facility. The cool thing about DNA, and the thing that I love, is a double helix. You split it apart, and you can read it. You can read it like a book. You can file it like a book. And you can build libraries. Once you build a reference library, you can come back with an unknown and match it. And that's the beautiful thing about DNA. And here's an example. And we do this at True ID. We build these libraries. We actually barcoded the fish of the world. Sounds pretty amazing, but when you have $150 million, you can do stuff like this. So we build these libraries, you have a can of tuna, and then you match it, and you see what comes back. Our latest research has shown that 65% of canned cat food that you feed your cats has CITES-listed rare and endangered sharks, mako and blue sharks in it. This is going to hit the news fairly soon. So we can go in and test the marketplace and see what's going on. It's a source of protein. It comes off the coast of South America. I'm not here to talk about sharks, but this is a reality. And you're going to hear more about this in the news. But it's the type of powerful thing we can do now. And I'm going to talk about how we actually do that and why it's important for the cannabis industry. So here's my team that I'm part of at the University of Guelph. We have the International Barcode of Life. We started it up. There's no other facility on the planet like this. We collect samples from all over the world. We work with Smithsonian Institute, Kew Gardens, Institute of Science and Technology in China. They all bring their samples to us, and we work them and run those samples in our facility. We have high-throughput robotic systems. These are our collection sites. Those collection sites are all over the world. You can see all these dots on the map. They show where we've been collecting. We've collected now over 7 million samples. And remember, every one of those samples is in a museum. We've extracted DNA. For many of those that are plants, we're doing metabolomic profiles now. We know where it came from, so we have this ancillary data for it, known provenance where it came from. This is particularly important information for building libraries for many commodities, including cannabis. So that represents 68,000 species of plants I have in my libraries. And here's how it works. Let's see if this pointer works. Oh yeah, it's going to be there. Ah, I found it. That's cannabis bud. Goes down. We take a sample of it. We extract DNA. We do a polymerase chain reaction, so we amplify the DNA. We sequence it. It goes into a bunch of sequences. These are the actual trace files. So these would be like HPTLCs for individual amino acid base pairs. There's the sample. We have a map. We have all the GPS coordinates. There's a colorized barcode. It all goes in the database. Remember, we do that 7 million times. We've done that. Okay, now you're thinking, okay, get on with it. Talk about cannabis. So here we go. Cannabis is fascinating. In my libraries right now, we work with 1,000 medicinal species of plants. By far, by an order of magnitude, cannabis is much more diverse. It's not just that cannabis has this breeding system, which is quite complex, gives rise to hybridization, a bunch of other mechanisms that makes the genome quite complex. It's quite an incestual plant, so a lot of breeding between siblings. And the sharing of the genome, it washes out selective sweeps in some parts of the genome. Other parts of the genome explode through mutations. So it has this high mutation factor, which gives rise to diversity. It's an evolutionary process. The other part of cannabis that makes it so diverse is it's very, very sensitive to phenotypic expression. That means the plant produces chemistry on very fine-tuned triggers. And you know this in the industry. So you're growing cannabis in a glass house, you change the lighting a bit, particularly light. I have like 4 PhD students right now just working on light and metabolites. What happens when we change the wavelength here? We have a whole expression of different chemistry. And the plant responds very quickly, minutes. It's actually totally fascinating. So as a medicinal plant biologist, I just find this amazing. This thing is a chemical factory. So for those of you who are working and you're growers and breeders, you create something. You've got the perfect platform. You're like a chef. So that's pretty amazing. The cool thing is in science is we can measure it. So I want to back up a little bit now and say, well, where did cannabis come from? So as a plant biologist, I need to always think about the range. Where did this thing live naturally? That's not where people collected it and they had ponytails and they brought it back and they grew it in their backyards or they grew it off hidden away somewhere in the forest. But where does it grow naturally? Where did it show up? And we do not have scientific evidence to really pinpoint this, but we have hypotheses. And we believe that it's over here somewhere in Northeast Asia. So it's somewhere up in the Himalayas, into Pakistan, into Tibet. So I have this project. I wrote this grant. It's this international grant between China, India, and Canada, of all places. But it was one of these grants where you could apply for anything you want, international collaboration. I said, I want to find the center of origin of cannabis. I want to collect all these plants, do all the genomics, metabolomics, do the evolutionary phylogeny, and show where it's found. And it's not like we haven't done this before. We've done this before for millet, and we're doing this right now for quinoa, or quinoa, you might know it as. And we're looking at the center of origin for it, and we're building all these tools for the industry. So we're going to do it again for cannabis. So we're doing whole genome sequencing, we're collecting all this data, but right now the hypothesis was that a long time ago, cannabis originated here. There was a speciation event. And this was thousands or millions of years ago. No, it's not thousands of years, this is millions of years ago. But the document is, this plant started to spread through time. And you can follow this history, and I'm not going to go through and map it all, but you can see how recent it is in North America. Basically, this sample has moved out of its original origin and has been bred in captivity as a clone. And in the cannabis industry, remember, I'm a geneticist, and I come from a university where it's an agricultural university. We do breeding programs. So much of what I teach is like fourth-year genetic breeding programs. And cannabis does not have a breeding program. It never has. It's like the early days of wheat 300 years ago when people were just starting to make crosses. So you do not have what we call an F1 or an F2 fixed trait breeding population. Getting there in hemp-based populations, but it doesn't exist right now. So a little bit of a background on the origin of cannabis. There's my best friend Raghu. Raghu and I are going to be, we've already collected up in the Himalayas cannabis, but we're going back in this project in August. We're going to be northern Himalayas into Pakistan. Yes, we're going to have bulletproof vest on. I have stories I can tell you about afterward where we had to hide from militants before. We're going to be up in Tibet. We're going to be collecting through China. Here's an example of the number of accessions. These are samples that we've collected through time. So in Canada, Canada has legalized marijuana. Canada has legalized edibles as of October of this fall, which has set a whole pile of balls rolling that I'm going to talk to you about. It's really why I wanted to come talk to you today. We're working with Jamaica right now to set up a registry. We're setting up the registry in Canada, 178 licensed producers times approximately 100 strains. Some have 200. So we have all these populations we're running right now. So we're building the registry in my lab for Canada, for Jamaica. We have all these public strains. We have wild type collections that we're collecting all over the world, including Brazil. And we have hemp populations that we get from you. I would love to get your populations. If you send them to me, no charge, I will mount them, label them, suck out DNA, and create a metabolome for you. So, for a limited amount of time. Accessories included. So, cannabis chip. First one, DNA. So, we've built a cannabis chip. It's based on, for those of you who are a little geeky in the crowd, it's got 400 genes in it, so it's the largest. It's got 60,000 SNPs. It's got the most single nucleotide polymorphisms. This SNP is where in the sequence, when you look at two closely related species, there's a specific mutation. So a change from an A to a C or a G to a T, which are amino acid base pairs. We are looking at chloroplast DNA and nuclear DNA, particularly chloroplast DNA, because that is the evolutionary process and the mechanism for chemistry. This is where the chemistry is actually coded for. And this is our phylogeny. These are all samples that we've run so far. And you can see in this phylogeny, this classification. So remember the old classification trees where you have all these branches, and one set of branches is called the clade. This is cladistics. So it's a classification, really math algorithm. So when you get a lot of samples, hundreds of thousands or millions, it starts to fold on itself, and we have this what we call a ratchet tree. It's circular. But every one of these lines is a population, and they show genetic similarity. And this is whole genome sequencing. This isn't parochial going in and looking at, Ribisco genes or looking at cytochrome C1 oxidase if you're kind of a gene head. Not these little regions. We're measuring the whole genome when we do this. It's kind of interesting as a geneticist. Back 15 years ago when they started to nail down the human genome, and it cost several $1 million. I can do your human genome for $950 right now. Nothing about your human genome that makes it simpler. I do mine for $950. That's all it costs right now, and I can do it in a day. It's pretty cool. So we did this for cannabis, and we started building this library. And it's not as if the genome there hasn't been a genome paper. There's been 4 papers published now on what they call the reference genome for cannabis, and one of them is in Science. It's just those genome papers were based on a very small population, Jamaica. One farm in Jamaica. And you would think, this is logical, you would want to collect it from the whole range, like that range map I showed you about. So what are we doing? We're collecting it from all over the bloody place and running all of these samples. And the most important population is your population. What's in the supply chain? So we're getting it from you as well. So when we run all that, this is what it looks like. And you can see that the indica and the sativa ones break out. They're actually quite different from one another. I've colored in red here to see where there's actually THC production that's coming through. And you can see that it's quite blended in here. This is the ancestral part of the breeding mechanism is really complicated in cannabis sativa. That happens. It's not clean. We can see all this roadside stuff that we find right here in Ruderos. It all is one species, cannabis sativa. I would argue, and we may actually break it into multiple species as a taxonomist. We'll see. I don't know. And then we have these hemp clades where we have low THC. But they show up in various places. It's not clean. Can we build a really good, consistent tool genetically right now? No, sorry, we can't, not statistically. So the other part of what I do is I have a degree and advanced degrees in math. Like I love numbers, and I love math. And I'm a classification mathematician. So when I look at this and I do the classifications, talk about p-values and probabilities and making a test, I can't make you a consistent test right now. I just can't do it. So But what I can do is look at multivariate space, and you're going to see a bunch of these as I go on today, and talk about variation. So I have school groups I go talk to, and they come in and they go, what kind of scientist are you? Like, you're a botanist, right? And I said, really, if we want to get down to it, I'm a scientist. I study variation. I measure things, and I study variation, and that's what's cool. Where did the variation come from? My model is plants. I'm passionate about why plants give off chemistry that gives us a healthy lifestyle and sustains life, and how we actually found that through time. But as a scientist, a pure scientist, I'm interested in variation. And as a mathematician, I'm interested in variation. So here's how we study it. And remember in the early days when you're in math class, and it'd be like an x and a y. And it's really simplistic. So you have an x variable, something that you're measuring lots of samples. And then the y is this thing you're interested in. Say it's the height of your hemp plants and its moisture. And as you add more moisture, they get taller. But you would think, well, there's got to be more things involved than height. So mathematically, you would think scientifically, that's a very simple model. There's got to be more variables. So we've left that behind when we get into complicated scientific questions. And we move into this box scenario, multivariate statistics. These squares and shapes and colors are all samples. If they're close together, they're similar. So whatever it is you're measuring, that dependent variable, they're similar. And what I'm going to be talking about today is their genetics and their chemistry, their metabolomics, which measures all of their metabolome, the phytochemistry they produce. You have two samples that are close together. In that space, they're similar. If they're far apart, they're different. And that's all you need to remember. Now, let's think about the hypotheses and the science behind this. Plants breed. They have a genetic code. So we have the genome. The genome codes for things. Makes us different from one another. Male, female, different color hair, no hair. For plants, it makes chemistry. It tells how tall the plant is, how it's going to deal with disease, so disease resistance. The part I'm interested right here, and we are in this room, I think, a lot, is the metabolome. What is the chemistry it's producing? How much CBD? When does it produce it? And then we have the environment. This is the mechanistic driver of it. Light, we know, is important for production of different metabolites for different parts of the genome in different strains in the population. This is the path through science that we need to explore. This is our experimental model that we work in. So we do that. So here's your reality. There are gaps in scientific knowledge that you have to deal with. There's misunderstandings. I come to these conferences and I hear people talk and I go, oh my gosh, that's not right. The first one is strain. It's a colloquial term. It's a common term. It's used in the industry. I totally respect it. The chefs who are the growers grow things that are real. They can taste, smell, smoke it. People experience it. Yeah, it's real. It has certain chemistry. Is it a taxonomic name that is recognized in science and can be defined and measured? No, never has been. It's not a genus species. It's not a division, class, order, family, genus, species. It's not part of that hierarchical of taxa that we have, taxons, and it never has been. So we need to be able to define a strain, and I have a way that we can talk about doing that. So we need to characterize that. We need to characterize it. Why? Because that's your commodity, and you need to have a consistent commodity through the supply chain. That's part of GMP. We need an identity test for it. So if you're going to be trading and moving products around, you need to be able to trace what it is. And if you have a thing called a strain and you can't measure what, it's a big problem. That was the number one key issue we dealt with for the last 2 weeks all through Europe. Consistent problems. Problem number one. And we had a number of prosecutory agencies. I met with the police high commission in Germany who want to be able to go in and police cannabis. It's like you can't even measure what, let alone do it in a validated model, let alone know the limit of detection and quantification. We need to figure all that out. Metabolites are not even characterized. When I first started measuring the metabolome almost 6 years ago in cannabis, there were 87 that were published in the literature, terpenes, flavonoids, on and on and on and on. We have 572 today listed in our database in our lab. And that's just in a few years. And many of them are new. Canoflavin A and B, we found it in a number of populations. I passed it off to Tarek Akhtar on our campus. He studied the molecular structure of it. He looked at bioactivity and a number of different arrays that he set up and found that it's 100 times more potent than aspirin in reducing inflammation, controlling pain. And that was discovered this spring, and we just patented it at the University of Guelph. We have rudimentary breeding programs. I won't be redundant. I already said that. Whoa. And the last one down here, we don't have a regulatory system. So one of the reasons I met with the British the German High Police Commission is they want to be able to police this, and we don't have systems in place to say what's illicit, what's licensed material. In Canada, I'm working with our RCMP, our Royal Canadian Mounted Police, police associations. We're building these libraries, and the question is, and we're building a registry for licensed. So the question in the test that I've built is it in the licensed registry, yes or no? If it's no, is it in the illicit material library? If it's yes, it's already related in the database to information. Oh, it came from Vancouver, and it was confiscated on this date by Hells Angels, this biker gang. And somehow it made it into the supply chain of food. That's actually a real case. So we have a sample. Oh, timing's good. Yeah. And we extract DNA, and we make a metabolome. And here's how the metabolomic fingerprint. So I just spent a few days with Bruker in Germany. They make these things called nuclear magnetic resonance machines. They make most of the MRI machines you find in hospitals. And it's basically a big magnet that's found here. And you run an energy signal through it. And when you do that, the molecules, the plant chemistry that's in there, that's held together by bonds for a fraction of a second, and I'll go here. So here are all the bonds that you see, the proton structure. This is proton NMR. These bonds break apart just for a fraction of a second. But they are so strong they come back. And the scientists that invented this are absolutely brilliant because when they come back, they go, aha, it's an opportunity to measure and recreate the structure. Because when it comes back, you can measure every one of these bonds in the protons. So you know the exact structure of the molecule. It's absolutely brilliant. So when you do that, you get these peaks. So this is strain 1 and strain 2. These are different resonance signals that you get from proton structure. They represent variation in the chemistry. You can see it's very different here from one from the other. I can use all this math that I'm not going to get into here. We can later if you like, which is called deconvolvulate. So I can actually figure out the proton structure and name that structure. And there's equations to do that. We can look at that structure and look at the variability amongst a pile of strains. And here's one of these ordination mathematical ordinations that's telling you the difference between 4 different strains. And you can see they nicely separate just based on their metabolome. And any one of these, depending when you look at it, may have 70 to 120 different metabolites in it, different chemistry. So it's not just THC, it's not just CBD. It's not just CBDA or B. It's not the terpenes, not the flavonoids. It's everything together. And what do you think's interacting with your body? For those of you when you put your hands up and you know somebody and are having a positive effect, do you think we can point to one thing in science and say, yeah, it's that, and it's only that? No, it's probably multiple things. Life's much more complicated. Science has shown over and over again, single mechanisms in the body do not exist. There's multiple ones. Some are more powerful than the other, but they're multiple. And often when they come together in multiplicity, they're more powerful. So here we are with peaks. Those peaks represent a bunch of metabolites. I can code them. I can make them into these ordinations, and I can start to classify different strains based on chemistry. Now I can answer the question, what? What is it you have? So here's a bunch of strains of cannabis, and they're all breaking out beautifully. And this is what the actual barcode looks like. Along this profile, there's a signal, and this is 1s and 0s, just showing presence, absence of different metabolites. And they're different as you look down. So you can see there's different chemistry. So we have this process now. We take a sample, we suck out DNA, we get part of the genome, just so we can start to build this library to use it as a tool over time. And the most important thing is we collect this chemical fingerprint, and this becomes a tool. So here's an example right here. I have the DNA chip. You can see there's variability. I can see chemistry variability. We match this in with what's actually on a product barcode label. This is Global Standards 1. And you now have a supply chain tool because this is a unique fingerprint for every one of your products that you put out, including finished products. So I can do this with an oil. We can do this with raw bud and produce a unique fingerprint for everyone based on the chemistry. And the beauty of it, this is the chemistry that's going to react with the human body. So we're doing this right now for human health trials. It's a mess. If you're a consumer and you're going out to buying it, this is the mess you deal with. It's not your fault in the industry because you didn't have a tool to make sure you have a consistent brand. We're giving you a tool right now to be able to do that. And for some of you, remember I did show a picture where it's really consistent. So for some of you, you're selling it, it's like you nailed it, you dialed it down, the variability. Remember, the plant's incredibly variable. You have to play with that. But you have a way to do it right now. And the key things you have to do for herbs of commerce and for GMP is identity and purity. You must show that what it is all the way through. Now you can measure what, and you must show how pure it is. Here's another example. Consistent product, same batch. So 3 products, same batch number, should be exactly the same. So these different colors are the different batches, and it's all over. These things are not the same. It should be a really tight circle right here. All the dots together, all the colors together. And you can see there's separation. There's no consistency in batch. I showed this to one of the big retailers that we're dealing with, like $28 billion a year retailer. And they looked at this and we can't launch this out. We can't launch this product. This has to be one circle. And when we have a circle, that is the circle of compliance for the product. I can make a math algorithm to say if it falls out from a supplier, it's non-compliant. If it's in the circle, you buy it. And that's qualified supplier, supply chain. And we're developing these programs for them. And it kind of works like this. We do that, we build it. This is actually a consumer app. Consumers can look at actually where it came from. So we're going to link the GS1 barcode for all products to a consumer app so consumers can look at it. The key thing I'm interested in, am I doing for time? Oh, I'm good. The key thing I'm interested in is right back to where I started, experience with plants. I want it to be real. Why did we start TrueID? 40% adulteration in natural health products. When someone buys echinacea to boost their immune, to deal with a common cold, they should experience what echinacea can do for you. It shouldn't be adulterated with alfalfa because you can bale it like hay. People do that, and they think there's no way to test it, and then as a scientist, they're like, no, I can build a test. We're going to stop that. In the cannabis industry, I think there's great potential for healing and helping people in society. But it doesn't help when you have wildly variable products that are going out or you have people that are mixing in different products. You need to figure out what it is that is going to heal people and then market out that product and dial in the variability. And people just didn't have that tool. For the people who are taking it, they should have some assurances really quickly on their phone, scan a QR code, and be able to come back and say, oh yeah, it was tested here, this is what you're getting. There's some accountability that comes back to them so they know that they can buy it. And then it boils down to their experience. They try it, and it works, or it doesn't work. So here's one thing we're doing with a company called StrainPrint. So we've hooked up with them. So we have TrueTrace, which is this blockchain traceability. We shoot data in from our machines, from TrueID. We can measure what. And StrainPrint's working with doctors. And they have patients. You know, doctors have the ability, have software that when they collect from a patient your ailments and you describe, you know, I've got a fever, and then they take a blood test, they take a urine test, all this data comes in. And it's not like they cook it up in their head like they used to 100 years ago. They put it in a program, and it comes back and it says, here's the pharmaceutical, here's the dose, here's all the information you need for the script that goes out. They don't have that for cannabis. Pharmacy's like, no, we're not doing it for cannabis. So this company, and there's several of them, have decided we're filling this niche. We're going to make this program for doctors. So now doctors sign up with them, and it's really a business-to-business thing. So the doctors and this company that have the software. Meanwhile, this company says, I'm going to also match up and provide a service to people, consumers. If you want to take cannabis, you want some guidance in what to take. And this includes hemp. And they go in and say, yeah, I've got these problems. This is my doctor. You know, guide me into what to buy, where in the market, where to buy it online, which company. And it kind of matches the two. Then it has this little app, and it comes back to you. And it'll come back, say I'm the patient, and it'll come back to me and say, Steve, you're taking it for joint pain because you're running too much. And it's like, yeah. Stop running. It's like, no. It's like, okay, take your CBD. It's like, take my CBD. It's like, it's noon. You're supposed to take your CBD, and this is the script that you have. You're supposed to take it at this potency and take it now. And, oh, by the way, Steve, on a scale of 1 to 10, what's your pain? It's like, yeah, it's a 9 today because I just ran 30 miles this morning. So comes back 20 minutes later. Steve, And it's on my app. It's like, and it's like, I think it's going to be my friend. And it's like, no, it's this bloody app talking to me and asking me, how's your pain? On 1 to 10. No, first of all, did you take your medication? Yeah. How's your pain? Oh, it's a 6. That data goes in. Maybe it's still a 9. It's like, yeah, that didn't work. It's a 9. But it comes back, it's a 6. So here's the beautiful thing, experiential data. It's like, oh, that's great. We are measuring the what? So we're getting the chemistry at the same time. And I have all these, remember, I'm a math geek, so I write all these little math algorithms, which is AI, and I have these spiders. And these math equations that go in, and they're looking for patterns. And I'm having patterns coming back saying, there's a real drop in pain, and a whole pile of these patients, this is experiential, this goes back to where I started. This is the beauty of the old and the new, what people always did with medicinal plants and what we're doing now is social media. So I have all this data that comes back, and it says, Steve, it's dropping off, and there's this particular metabolite that really people don't know about. It's a flavonoid, and I go and look at the protein structure and go, yeah, it's bioactive. We don't know anything about it, but it's reducing people's pain really quickly. And we're getting this information. We have 1.8 million users right now, patients, giving us data. And we're measuring right now the metabolome. And in my lab, I have this robot and shooting it through. I can do it really cheaply, really fast. So we're running all these samples, and I'm getting all this data. Now, I'm getting back negative effects too. People are coming back because in that app, it comes in, phone goes off and I look at it. Do you have any negative effects? Like, yeah, I got a headache. And what if, 180,000 other users that use that strain are getting headaches and I see the exact same metabolite showing up? It's like, it's experiential, it's exploratory, and it gives rise to hypotheses. And now I look at the proton structure and I go, oh, that could be, and then we do studies on Sprague-Dawley rats and zebrafish models and eventually humans to say, yeah, you don't want that strain in the supply chain because it's negative. Some cause paranoia. That's not a CBD thing. This is more in the bioactive THC realm. But we've been working with Health Canada, and I'm done at 10, right? I'll see much more. I'll see if I share that study. Yeah, here's the supply chain stuff. Here, we're setting standards right now, so in the background, we're only standards organizations, and we're bringing licensing and standards, so I won't bore you with that. And before we get to questions, I'll share this story. So I'm at my lab in Halifax, and some people in the crowd have actually been there and saw the zebrafish. So the zebrafish is really cool. So you take chemistry, you put it in the water with the zebrafish, and they use zebrafish models because apparently their brains are very similar to ours. So these neurological scientists or neuroscientists are studying zebrafish all over the world for pharmaceuticals. So we're doing it for cannabis. And we're trying to understand paranoia. So, and this is way out of my science thing, but so I'm there as a science geek, and I go, so how do you know the zebrafish are paranoid? So they have cameras and probes on these zebrafish, and the zebrafish are moving through. So apparently, if you're an ichthyologist, like you're a fish biologist, zebrafish have a pattern. They're kind of like old people. Like they get up in the morning, and they go over here and do this, and they go do that, and they go do this, and they go to sleep, and they get up in the morning and go do this. And so you then put in this particular metabolite that we think is associated with paranoia, and the zebrafish get up in the morning, and they go over and do this, and they go back and do this, and they stop, and they go back and do over there and check to see whether or not they actually did that. And then they go over here, and then they go back, and they're going back and forth. They're stuck in a rut. It's like, oh, that's a paranoia mechanism in psychology, that you're kind of going back and checking all the time. So that's one of the mechanisms. It's like, no way, zebrafish are paranoid. So I'm going to stop there. I'm around for the next couple days. If you want to chat, I'm available. If you want to get a hold of me, I'm at the University of Guelph. Actually, we can do something. I'm not all that technologically good at phones, but I'm going to do this right now. If you have LinkedIn and you go to LinkedIn, where the heck is this? Yeah, there it is. And you go to the little button that says My Network, and you look at the bottom right corner, and I'm clicking on it right now, and you click on it, it'll say Find Nearby. You will find me. And we'll connect on LinkedIn. And then you can ask me any questions. So I'll leave that open. Oh, yeah, look at that. I got someone already. Cool. Yeah. So I usually do this at conferences, like, and they'll come in. And then I can chat with you, and I'm kind of like work all the time, so I'm happy to chat. I'm happy to educate. It's what I do. Okay, thank you. So are we doing questions, or do we have time? Probably. Yeah, good.
So if I'm buying CBD on the open market for my product, how do I incorporate your testing to make sure that I'm and I'm working through you, I guess I don't follow how I.
As a consumer or a manufacturer?
You talk about me?
Yeah.
I'm
Or
buying CBD
yeah.
from manufacturers.
Okay. Yeah.
And then resell.
Yeah. So you could send it to us to do, or we're going to license labs, and that company that I'm dealing with in Germany that actually makes the gadgets, we're going to have those in the labs, and you're going to be able to do it, and it's going to cost you approximately, just to give you an idea of scale, $150. It shouldn't cost much more than that. And you will get this chemical barcode for it. Now, we'll have genetic tests, which will be a proxy for it. And I didn't talk about this, but when that genetic database gets really big, and I have the really big metabolomic database for the same plants that I'm collecting, I can now do the cool math where I associate the two. So it's called canonical correlations. And I can do what's called proteomics. I can make a probe for the genome that codes for that chemistry. But right now, what we can definitely do is just give you a metabolomic fingerprint. We're just calling it a cannabis barcode. So you can get a cannabis barcode for $150 right now, and run your product. And then when you go run it again, it's consistent, and that's compliant then for GMP. You should be able to do that's called verification. So you're verifying what you bought as it goes through the supply chain. And the idea is to make it a consistent product. Do I want to make it expensive? No. Why are we certifying labs to make sure they're ISO 17005, ANSI accredited, and they're going to follow our validated methods, and we're going to put in a price cap on it and saying, we're not sharing our libraries with you if you're charging too much. Because some labs will be like $1,500, make a big margin. It's like, no, you don't get licensed. This is for everybody to use. That's why we're doing it. Does that answer it for you? Yeah, so an interim thing, you can just send it up to me, especially if it's CBD, no problem. You should be able to do it. And then we're going to be licensing labs in every state. We're just in the process of licensing. We're rolling out the certification for January 1. So contact me, and I'll be able to set you up for the new year. Yeah, so yeah, for the new year, depending on what state you're in. But yeah, and I'm kind of flying all over the place, going into labs going, oh, I didn't see that. It's like, no, you don't get certified. Yeah.
HPLC testing.
Yeah.
I walked in when you were just talking about that.
Yeah.
HPLC testing, are we, right now, would it be accurate to assay total?
Here's the problem with HPTLC. So here's a chemistry lesson. HPTLC measures a specific chemical. Peak, fluorescence. You need a reference material standard that you usually buy from US Pharmacopeia or NIST. They don't have any. So you have the standard, you have the unknown, they're supposed to match. Now they have no math to say what's a match and not a match. I've been after them that for years. It's like, get out of here, new master. But that's one chemistry. When we do metabolomics, we're looking at everything that's there, not just one. So when you pick one, you have to ask the question, well, why did you pick that one? So when I go to the regulators and they go, we're measuring 10 terpenes for, blah, blah, blah. And I said, the terpenes, when you make products and you go through steam treatment, blow off in 90 seconds, they're gone. And when we measure with metabolome and we look at terpenes, when I measure the terpenes and I look at the structure, they are spruce and pine terpenes. They put industrial terpenes back in, which we're not supposed to be eating. They're not fit for. So I'm looking at it going, oh, this isn't good. So HPTLC is very limited right now. We don't have standards, and it's a very small view of the chemistry. So, and if you pick one thing, you better have a good reason for why you pick it and have all the science that underpins it, and you don't. So.
Not just HPLC regarding just assay, like total potency of the product. Would we consider that to be accurate?
No, you have no reason. What are you measuring for potency? Which chemical? So as soon as you say a chemical, then you need to know which one. You need a reference material standard. You need limit of detection, limit of quantification. Nobody's done that work. Nobody. So when I walk in, I'm in Holland, it's like, yeah, we got this figured out. And you think they would. And I said, did you do all these things? They go, we don't need to, because it's like,
so if I have
this is wrong.
Doing HPLC assay testing right now, it should not be trusted?
Yeah, I would say, what are you doing? And why are you doing it? I would ask them those tough questions. Do you have a validated model? Where do you get your standards? You should be asking those questions, or you're getting taken to the cleaners. Hate to say it. And I'm rather blunt now, just because just clean up the industry. We're going to give you a tool. It's going to be validated. There's going to be certain ways of doing it based on standards. And it's Wild West right now. People are just hanging up shingles, and they're just charging these outrageous prices. It was part of a study in the US. We measured across 10 different commercial labs, licensed commercial labs, and some big ones. And we measured CBD and THC. We found 37% standard deviation. That means when it was supposed to be this, there was this big amount of variability around it. It should be less than 1%. So,
Finding about 37% barcodes.
yeah, that shouldn't happen. I know. FDA is going like, oh my gosh, what's going on? It's like there's no valid, they're not ISO, they're not ANSI, they don't have any validated methods, they don't have any reference. It's no wonder. I could give all kinds of hypotheses why. Other questions? I've been doing this a long time, so, and we do it for so many different food ingredients. So jumping in with cannabis is just like one of the other 1,000 that we do right now. Yeah?
So you showed a digital, like a barcode, which describes distinct rows of lines.
Yeah.
So that means that at some point in time, there was a transition stage between the 0 and 1. I'm assuming that's the limit of detection.
Yeah, that's brilliant. So that's good. You know, I threw out the zeros and ones now like 2 weeks ago because I went back to my team and we're just getting ready to do the standard for it. And I said, let's use the resonance because the resonance is the potency of that particular metabolite that's in there. So that colorized one that I show you, I just got the other day for my team when we're running a whole pile of CBD products. So I'm going to move away from the zeros and ones. I could simplified it down, but you lose too much information. So having the actual resonance from the chemistry gives us a level of potency at that particular resonating spectrum. We calibrate that through mass spec, known proton structures. And those standards are being built by metabolomic centers. Like there's one at University of Alberta in Canada. There's one in Germany as well. They're very detailed structures. So that becomes, yeah, so that's proton physics that we use as our standard for it. Now, we got this dialed down. We got this nailed. But those are really good questions. Anybody else before they kick us out of this room? No? We're all good? Okay, thank you.